Okay, so I was presenting my laptop downstairs with a table, so I could put my laptop with my notes. Okay, we'll see if this works. So, actually, I'm just going to do it with my notes. Fingers crossed I don't forget anything. Yeah, so this is about the tools and skills needed for digitizing collections. So we're all doing a really good job in digitizing our collections. Some of us have managed to do most of their collection. Some of us are still working to either complete it or to get started. But what I wanted to do was talk about some of the skills and the tools that are needed to do that and how we can work together on those. So, I'll just do it from here. Yeah, so the kinds of things I'm talking about are where we've got the tasks that are involved, we've got the tools, we've got the different roles involved in digitizing collections, we've got the resources, skills, sorry, it's got a little bit distorted. But, and then we've got the workflows that are pulled together for the different parts of the work. And so what I wanted to do was maybe just start with looking at an example of a workflow, what we're kind of talking about. So this is just an example of a workflow that we have at the Royal Botanic Garden, Edinburgh. It's a kind of overview, and it's very much based on that super paper, the Nelson et al., the 5 task clusters of digitization. And that's been a really good basis for a lot of the development that we've done. So this is the kind of workflow that I'm talking about. And then what we have as well is within the workflows, we've got things like the digitization roles. And there's a number of, I've just put some examples in here just to kind of show you what the kinds of things we're thinking about. And then the tools and resources, the kinds of things we're looking at. I mean, there's quite a lot in digitization. I think it's not always recognized, the complexity of the things that we're doing. And also the skills in software that are needed, especially with such a fast-moving area, that all of this is becoming more complex, there's more skills needed all the time. So thinking about how we can connect these things together so that we can connect the workflows, the tasks, and think about how they feed in together and what we can do to work collaboratively to bring the things that we need together and to make them available and accessible. So I've just taken an example here, which is for image capture. It's just one part of the whole process, part of the workflow. And so with this, we've got some of the tasks that are needed to do image capture. We need to get specimens out, we need to take the image and get them back in again. There's tools and resources that are used for that. There's skills and training that are needed. Another example role that we can look at is the digitization coordinator. And again, you've got the different tasks, tools, and skills that are needed for that role. So I kind of had to think about, well, what's out there, what's available, what can we be thinking about to help us? There's the carpentries. And we've been talking about building a specimen carpentry earlier in the week. So the whole carpentries thing, I think, is something we should be looking at. There's another thing that I think maybe not so many people have heard about. It's called the Turing Way. It's from the Alan Turing Institute in the UK. And they've come up with some really interesting structures. So they've built this big guide. They've got guide for project design, guide for data management, guide for collaboration, communication. They've got a huge, huge handbook that they've built as a community. And what they did was, within that, is they also then came up with these pathways. The pathways are basically different roles. And then they've taken all the different bits of the handbook and they've pulled out all the different bits that are relevant to different pathways and made that into a kind of resource for people to use. So if we take both of those, and then we take something like Obsidian, kind of mind map, kind of second brain kind of thing. Just one question. How many of you here have heard of Obsidian? Okay, small number. I would recommend having a look at it. It's a lot of fun. And it can do some stuff. So I was taking those things together, and I thought, well, what can we do? So first of all, we can create a vault in Obsidian, which I did. And then we can create guides. So for each of those tasks that we have in digitization, we can create guides for those. And we can break them down so they're in small chunks. And it could be like checking the camera settings, for example. And each of these could be done either specifically for a particular institution, collection, or it could be done, some of these would be quite general, depending on what the thing is. So for specific things, it probably makes sense for each institution to create these little guides themselves. But there's also a chance that you can save PDFs into your vault. So some of the PDFs might be things like the Atlas of Living Australia, the guidance that they've got. And there's a lot of things like this out there. It's really, really good stuff out there. And it'd be really nice to make it more accessible and bring it together. You can create pathways. So these can be embedding into internal links so that all of those guides that you've created in the vault, you can then bring them together. You can pick and choose which ones you bring in. So here I'm creating a pathway for a digitizer for image capture. So I can bring in some of the internal guides that I've created, and I can bring them into my pathway. And I do that just by putting the link in, and it actually brings the resource in. It's embedding a kind of active link, if you like. So if I update the original guide, the updated version will appear here. You could also, I can embed external links, and this is a lot of fun. So with the external links, I can use some of the resources that are out there in websites. And I can then, so there's, looking at the, some of the resources here, which is the DISCO digitization guidelines. I pulled that as an external resource into here. So I can see it, I can manipulate it and use it within the Obsidian. And you can see, so as I go down my pathway, I can come to a particular guide, so the DISCO digitization guidelines, and I can scroll inside that, and I can click on links and things in that as well. So it's kind of active inside my pathway. And then it also gives you this option of visualization, so you can see how all the different things that you've created relate to each other. So I'm just winding up now. So that, thinking, I mean, this concept of building these pathways, similar to the Turing Way, how they've managed to do that. I think as a community, it's something we could look at doing. There's so much stuff out there. We could do this as a community and make the different guides available for people. I don't know if Obsidian is the right platform for it. It was something I could do. I thought I'm going to try it. I'm going to explore it. There's lots of questions about how we do it, and also how it should connect to the carpentries and the Turing Way. Do we want to connect to existing things? Do we want to create our own specimen carpentry? And could this be part of that as well? So, food for thought. If anyone wants to see it, I've been kind of like having fun on my laptop. So if anyone wants to see it, I'm happy to show it to you. And that's it. Thank you.
Thank you, Isabel. So we don't have time for questions, but maybe Good afternoon, everyone. Is that it? Yeah. Okay. Good afternoon, everyone. My name is Sidra Ikram. I'm from Cube Gardens in London, and my project, my presentation, is also on digitization. Mine's slightly a bit more generic overview, so hopefully I won't repeat too much from the previous one. So Kew has one of the most diverse botanical and mycological collections, but historically it's not been very accessible for people. At the moment, if you want to come and see specimens, you have to fly all the way to the UK, which is not obviously possible for everyone and not very environmentally friendly either. So we wanted to make sure that we get our data out there, and so it's accessible to not only scientists, researchers, students, but also the public as well. So we decided to undertake a large-scale digitization effort, which is a 4-year project, which we started in April 22, and we're finishing in March 2026. It was fully funded by the UK government, and we were given a £30 million budget. So within the digitization program, we actually have 3 major components. The first is the imaging and transcribing of our 8.5 million herbarium and fungarium specimens. The second was to create a central database. So apart from the herbarium and fungarium, we also have other scientific collections. And we wanted to have one sort of central space where we could put all of our data and information. So this was the ICMS project. And the third is to create an online platform where we can then put all of our images and all of our data online for people to access, and that's called our data portal. Next slide, please. For the next couple of slides, I'll just focus more on the digitization aspect. Next. So before undertaking a really large project, we've obviously had to do a lot of planning. We had to write a business case which outlined the objectives that we were going to achieve, what the targets were, and also potential benefits. For example, whether we wanted to see an increase in citations and publications as a result of our work. We had our own project board set up to make sure that we were on track. We have meetings with them every month, and they grill us quite heavily. One of our biggest questions was whether to do this work in-house ourselves or to do it externally, and we kind of did a hybrid model. So we had Max Communications, who are an external company. They've been doing the majority of imaging and transcribing for our herbarium specimens, but our in-house team, where we've recruited lots of staff, over 100 in total, they've actually been doing the imaging for fungarium and other special collections. Apart from this, we've also had to do a lot of procurement. We had over 45 camera stations set up, so a lot of lighting, equipment that was needed to be procured. And in some spaces, we actually had to install IT infrastructure. We didn't have network points in some parts of our herbarium because it's quite old. So there was a lot of pre-work that needed to be done first. Next slide. Next, I'll talk about some of the teams that we have, slightly repeating from our previous presentation, but we have digitizers who did the imaging and the barcoding and minimal data work. The transcribers were the ones who actually typed up all the writing that you find on specimen sheets. We had a quality assurance team who were able to then check all the images and transcriptions, not only from our supplier, but also our own staff as well. We had a team of data managers who then clean the transcriptions to make sure that it's in the right format so it can go into our central database. We had a project management team, which I was a part of, which, apart from having lots and lots of meetings, was just to make sure that our staff and our suppliers were all on track so that we can finish the project in time. One thing I've actually left off is we also have some volunteer teams that work with us as well, but I forgot to put them on there, sorry. And obviously, we've had a big impact on our IT finance teams at Kew as well. Thank you. Next, I'll just talk about some of the lessons that we've learnt. So some of our challenges we've had, the first was illegible handwriting. Some of these specimen sheets are hundreds of years old. The writing style is very different. Some of the location names may be outdated. Some of the language may not be appropriate anymore. So there's been a lot of work to try and sort of decipher that. We've also encountered multi-specimen sheets. So as you can see on this image, I believe this one has 8 specimens on here. So we found some sheets that actually didn't have the right number of barcodes and the right assignment, so we had to do a lot of that work. I'm going to ask my colleagues, I think we've got a sheet with 20, what was the biggest number? Something like 20 specimens on one sheet we found. I think that was our biggest one so far. Thank you. Another thing we've really recommend is engaging with your stakeholders, so not only your staff, but also the other wider people that work with your collection. So for us, it was curators. We had to negotiate space with them. We had to kind of persuade them that we are going to take care of your specimens, and I think they've really come around and really happy that we're kind of preserving their lifelong work. So that's been really important. The next 2 points, if possible, if your budget allows, I would suggest to stagger your recruitment. Just the way that our government kind of gave us budget requirements, we had to hire a lot of our staff quite soon in the project. And I think one of the issues with that was not necessarily all of our standard operating procedures were ready then. So that can be a bit tricky. So if you have that flexibility, I would suggest making sure that you have your SOPs ready and then recruit in that kind of fashion. Another thing that we'd recommend is to map out your targets and key performance indicators. So not just on a yearly basis, but literally a monthly basis. It keeps us on track, and also to ensure that you give yourself a buffer, because not everything goes to plan. Some things take longer than planned. So we gave ourselves sort of a 6-month buffer that we hoped to finish before, so that in case things take longer, we can still finish in time. Engaging with your organization's communications and PR team can be really helpful. It's something that Q didn't do before for specific projects, but they have for ours, and we've managed to bring in some funding specifically for us. Last one's a bit cheeky. If you have the time and resources, count your specimens beforehand. It wasn't something we had the time to do, so we had to estimate and guess, which was okay in the end. We've managed to get around the right number we thought we had, but it could be a little tricky. That's just a very, oh no, it's fine. It was just a sped up video of how the imaging is done in our stations, but we can go next. So as of the 10th of October, we've now imaged 7.9 million specimens. We've transcribed 5.5 of them, 5.5 million, and we're on track to complete by March 2026. Our ICMS projects, the Central Management System, we completed that earlier this year in March, and all 12 of our scientific collections are now in that central database. We actually used an external supplier called Earthcape, who are based in Finland. They helped customize their system for us, so it's almost like a bespoke solution for us. Our data portal has been launched this year. We're calling it in a beta version. We're still sort of working on it, improving it. I believe there are about 6.71 million specimens currently on there. There is a link. If you have a chance, please have a look and give us some feedback. We're still in the process of upgrading that. And I guess the last question is, now that this project's about to finish, what can we do next? Q does have a lot of other collections, including seed, economic botany, illustrations. So we are going to pitch that in the future, if Q has more funding available, that they do look at digitizing their other collections as well. Next slide, please. Just want to finish off by acknowledging that this was a huge, huge project, and I think we're quite proud of the fact that we are on track to finish within budget, which is quite amazing. So yeah, it takes a lot of effort, not only your own staff and your own teams, but also your suppliers. We've had really good sort of relationships with Earthcape and Max, so I think that's quite an important part of our journey, so thank you.
Nice piece of work. A lot of money you got for doing that. I want to ask if you are going to digitalize all what is in the Marian North Pavilion. 800 pictures from all around the world, goods and things.
So the Marian North collection is a collection of artwork by Marian North, who did a lot of traveling, I believe in the 1800s, if I'm not wrong. Okay. Okay. Yeah, she did solo traveling all over the world, which is a really amazing story. And we have a lot of her artwork at Q. I don't know entirely whether that would be digitized. Some of it's actually on the walls. But I can find out and I can let you know if you'd like. Oh, sorry, Richard might know, yes. I think
So there are also some questions online. Okay. So if we have time at the end, we'll do it. Otherwise, I encourage you to go online. And we'll just give our microphone to the next speaker, Jorge, who's going to talk to us about biodiversity. We have the implementation of biodiversity data management system. In Spanish. So actually, translation.
If you need the QR, here.
Yeah.
It's here. ¿Y esto funciona? Acá. Perfecto. Bueno, hola, ¿cómo están? Bienvenidos. Yo soy Jorge y les vengo a conversar sobre la experiencia que tuvimos en la empresa, en el diseño y la actual implementación del sistema de gestión de datos de diversidad con lo que trabajamos. Ellos son parte, no, me equivoqué, botón. Ellos son parte del equipo que colaboró en el diseño de sistema de gestión de datos, y les cuento un poco de la empresa. Somos una consultora ambiental que se dedica a la evaluación de impacto de proyectos y que trabaja con datos de biodiversidad. Trabajamos con altos volúmenes de datos de biodiversidad de distintos componentes y tenemos hace 2 años un equipo de datos que gestiona y gobierna el trabajo con los datos. Básicamente, y no tengo pruebas de esto, pero somos la única consultora que tenemos un equipo de datos. Como les digo, no tengo pruebas, pero tampoco tengo dudas de eso. Y además contribuimos a GEDIF, actualmente con 13.500 registros más o menos. ¿Cuál es la situación? Se nos encomendó al equipo de datos diseñar un sistema de gestión de datos con base en este problema. En general, las empresas que trabajan con datos de biodiversidad no tienen un sistema formalizado para trabajar con sus propios datos. Y nosotros tampoco. Más bien, si bien teníamos un orden con el trabajo con los datos, no lo teníamos formalizado. Entonces, con las integrantes del equipo, Stephanie, Soledad, Francisca y Jorge, yo, fuimos a un bar y nos pusimos a trabajar orientados en lo técnico porque somos técnicos, y evaluamos el estado actual del trabajo con los datos y definimos con todos los equipos que pertenecen a la empresa también el flujo de datos actual que teníamos y el flujo que queríamos llegar a tener. Básicamente el contexto es que tenemos un volumen de datos creciente que abarcan tiempo y espacio, que pertenecen a diversos componentes de métodos variados y de productos diversos. Y la orientación del diseño se basó en básicamente ordenar los datos, asegurar su trazabilidad, acordar criterios, mantener al menos la calidad, estandarizar formatos y, en definitiva, siempre ponernos de acuerdo en cómo trabajar. Teníamos que ponernos de acuerdo. Ese es el alcance del sistema de gestión de datos que finalmente quedó. Eso lo voy a comentar al final. Como les comenté, lo más importante era ponernos de acuerdo. Entonces, luego de revisar varias fuentes, obtuvimos la primera versión de nuestro sistema de gestión de datos, que se constituyó en un documento con todos estos ítems. Fue diseñado a partir de nuestro conocimiento técnico y con varias referencias, pero el problema es que sentíamos que no era amable con el usuario, no comunicaba de manera efectiva. Y lo que nos pasaba era que nosotros sabíamos lo que queríamos hacer, sabíamos lo que queríamos comunicar, pero no sabíamos hacerlo de manera efectiva. Entonces recurrimos a una asesoría externa de una profesional de la comunicación, Rida Quiroga, la que nos orientó en establecer un documento con comunicación efectiva para personas y con un formato amigable. Y además complementamos nuestra propuesta con estos 4 lineamientos, orientado al trabajo con datos, las consideraciones a tener cuando empezamos un nuevo proyecto, cuáles son los canales de comunicación y cómo comunicarse con nuestro equipo desde los otros equipos, y también las buenas prácticas asociadas a la denominación de archivos. Entonces, la versión 0.1, y con unos últimos ajustes y detalles formales asociados al sistema de gestión, llegamos a nuestro sistema de gestión de datos actual y estamos en proceso de implementación, y les comento sobre esto. Básicamente, posee un formato amigable, tiene un lenguaje simple y cercano, tienes elementos independientes pero todos interconectados, y lo formal está separado de lo técnico. Tenemos un documento que habla del alcance del sistema, un documento que habla del flujo de datos y los procesos considerados en él, los lineamientos asociados al trabajo con datos, los aspectos a tener en cuenta al iniciar un nuevo proyecto con datos, un sistema de asesoras, no la chunto, ahí está, perdón, cómo hacer solicitudes al equipo de datos, este es formal, un mapa de procesos sí, porque es algo formal, eso no lo vamos a ver. La definición y el uso de estándares como Darwin Code, y ya lo teníamos incorporado de todos modos, procedimientos y herramientas. Bueno, y les cuento una anécdota. Como buenos técnicos, nosotros partimos armando nuestra manera de trabajar, y ya la teníamos lista, pero lo último que diseñamos fue justamente esto, el alcance. Como buenos técnicos no consideramos lo formal. El alcance, en definitiva, del sistema de gestión de datos es una herramienta para orientar la gestión de datos dentro de la empresa. Provee de estándares, formatos, herramientas, procedimientos y criterios también. Y también abarca algunos alcances específicos asociados al tipo de datos, al tipo de usuarios, a su interoperabilidad, a los canales de comunicación y otros aspectos. No pretendo que vean este flujo, solo quiero que lo vean de manera macro. Este es el flujo de nuestros datos y hay dos líneas. Arriba tenemos el flujo que viene de donde vienen los datos de terreno, donde hay una etapa en que el equipo del componente flora o fauna trabaja sus datos, y hay otra etapa en la que al equipo de datos se nos solicita distintos productos, que son los polígonos con borde redondo en verde. Y abajo tenemos otro flujo de datos que tiene que ver con datos que no tienen una estructura clara, que vienen de otra fuente, y que tenemos que nosotros gobernar eso junto con el equipo encargado del proyecto. Yo no fui. Y ya. El lineamiento para el trabajo con datos tiene esos 7 orientadores. Básicamente es el orden con los datos, la trazabilidad, la precisión de estos, la completitud, la coherencia, su consistencia, su confidencialidad, y finalmente creemos que una de las cosas más importantes es la colaboración entre los equipos. El sistema de asesoras también es algo que implementamos para que cada integrante del equipo de datos preste asesoría a cada equipo dentro de la consultora, a Flora, a Fauna, a Limnología y otro equipo, a Estudio y Modelación también. Y los estándares, procedimientos y herramientas básicamente orientan en el uso de estándares, orientan en los procedimientos a usar con los datos, a su documentación y al desarrollo también a través de scripts en R y en Python, de validadores, de generación de figuras y análisis semiautomáticos. Y finalmente, los aprendizajes que tuvimos en todo este periplo de 1 año y medio se orientan en que la implementación y el diseño del sistema de gestión de datos es continuo, que es algo que aprendimos a la fuerza también acá, que un producto mínimo viable es mucho más, es mejor implementar un producto mínimo que uno ya tiene diseñado a esperar a terminarlo completamente. Necesitamos siempre una comunicación permanente, fluida y clara, porque, y esto es lo más importante que creemos que obtuvimos de aprendizaje, que trabajamos con personas, con conocimiento, experiencia, herramientas y procedimientos diferentes. Y que cualquier proceso de este tipo, como la automatización de procesos, debe incluirlas dentro del proceso. Deben estar no solo preparados, sino también en conocimiento de cómo funciona todo el sistema. Finalmente, quiero agradecer a Ciencia Ambiental, a cada integrante que participó en este proceso, que no salieron todos, lamentablemente, y quiero agradecer también a al equipo de administración de la empresa y especialmente a Paola Moneta, que sin ella no estaríamos acá. Y también a agradecer a integrantes del Ministerio de Medio Ambiente, Lacey Amaya y Victoria Arevalo, que nos orientaron en un inicio con nuestro diseño. Muchas gracias.
Very much.
Thank you.
If you have a really quick question, you can take it. Otherwise, we'll just move on to the next speaker, and you can ask questions later. Okay, thank you. So Katarina is our next speaker. It's also a presentation in Spanish.
So, hello everyone, my name is Catalina Merino. I'm coming from the Natural History Museum of Chile. And I was planning to give this talk in English, but since we are in Colombia, I think it's only fair to let my Spanish accent to have its moment too. So, I'll keep going in English, in Spanish, sorry. So, this is a work being carried out with me, conmigo y mis colegas. Se titula Curando los registros de corales y trata sobre hacer un análisis de los sesgos, vacíos y también desafíos que tienen las colecciones de historia natural. Entonces, acá tenemos un flujo más o menos generalizado sobre cómo es el proceso de colecta de ejemplares de las colecciones biológicas. Tenemos que tener súper claro que las colecciones biológicas son infraestructuras científicas que nos permiten hacer análisis muy importantes, son la base para estudios ecológicos, evolutivos, como también genómicos. Entonces acá tenemos un ejemplo, por ejemplo, de un coral siendo colectado, siendo en terreno, donde tomamos una serie de muestras que luego llevamos al museo y centralizamos en un sistema en un software de manejo de colecciones. Acá es importante tener claro que en el museo es un proceso de generación de conocimiento continuo sobre las colecciones biológicas, entonces son datos que se van generando a medida que las vamos estudiando. Y bueno, lo que solemos hacer es estandarizar esta información utilizando el estándar Darwin Core y luego poner toda esta información en agregadores de datos como lo es GBIF. Pero bueno, todo este trabajo tiene una serie de desafíos importantes, principalmente vacíos y sesgos en los que son tres componentes principales, como análisis temporales, geográficos y también taxonómicos. Entonces, tener en consideración estos vacíos es urgente, particularmente en ecosistemas como los marinos y en ecosistemas aún menos estudiados como es el océano profundo. Entonces, para hacer este análisis, nosotros utilizamos los corales pétreos, o también llamados corales escleractíneos. Decidimos utilizar estos corales porque los consideramos como un caso de estudio ideal de colecciones, porque tienen una larga data de colecta, como también son ecológicamente fundamentales, y también tienen una alta visibilidad, ya sea científica como pública, o sea, son especies icónicas, a quien no les gustan los corales, ¿cierto? Entonces, este estudio provee el primer análisis como una especie de síntesis de las colecciones de historia natural de corales. Cabe tener en cuenta que el análisis solo lo hicimos para especies recientes y solo con los datos disponibles en GBIF. Entonces, este es un flujo que muestra cómo fueron los procesos de filtro. Comenzamos con buscar escleractíneas en GBIF y consideramos todos los registros de basis of record removiendo los especímenes. Partimos con más de 1 millón de datos, luego nos quedamos solo con aquellos datos que están identificados a nivel de especies y ahí perdimos una gran cantidad de datos, ¿cierto? Luego tuvimos que volver a hacer un análisis utilizando worms y una base de datos que se llama Paleo Database, para volver a rescatar especímenes solo recientes. Y aquí también tuvimos una salida importante de datos, y claro, la salida más importante viene cuando consideramos solo datos que se categorizan como especímenes preservados en GBIF. Y luego removimos los duplicados y aquellos registros que no tenían números de catálogo. Entonces nos quedamos solo con 176.000 datos, en los cuales hicimos este análisis temporal, taxonómico y geográfico. Entonces acá podemos ver algunos de los resultados. Esto es las colectas de corales a través de los años. Esto es el número de registros. Y podemos ver que, por ejemplo, en la década de los 70 hubo un gran aumento en la colecta de corales. Y luego viene este descenso hasta la actualidad, en la que ya no estamos colectando tanto como antes. Y acá separamos según institución. Entonces podemos ver que, por ejemplo, en la década de los 70, el Queensland Museum se lleva más del 40% de los datos colectados. Pero también podemos darnos cuenta que instituciones como el Smithsonian tienen un registro a lo largo del tiempo que es bastante estable. Esta caída en los datos también la podemos asociar con la incorporación de los corales en CITES, que es este convenio que regula el movimiento internacional de especímenes protegidas. Y bueno, en cuanto a la parte taxonómica, los resultados son más alentadores, ya que tenemos un 92% de cobertura taxonómica en las colecciones de historia natural. Aquí tenemos las 1.561 especies de corales representadas en las colecciones y el número de registros. Pero algo interesante acá es que solo el 8.6% de las especies engloba al 50% de los récords, de los registros. Entonces, está como el sesgo súper grande a que unas pocas especies tienen una gran cantidad de registros. Y por lo contrario, tenemos más de 1.000 especies que solo tienen muy pocos registros. Entre las especies más comunes tenemos estas tres. Y bueno, si analizamos esto ya en términos de cuál es la relación simbiótica de los corales, nosotros podemos dividir los corales en dos grupos básicamente. Uno son los zozoantelados, que son aquellos corales que viven principalmente en el océano profundo y que no hacen simbiosis con las algas. Y por otro lado tenemos los zozoantelados, que son los que sí tienen esta relación simbiótica con algas. Y tenemos algunas especies que pueden ser o zozoantelados, pero acá lo que nos damos cuenta es que también la mayor cantidad de registros para ambas categorías taxonómicas es tener muy pocas especies con muchísimos registros. Bueno, otra cosa interesante acá es que de las especies que no encontramos en las colecciones hasta ahora, que son 141, se dividen casi en forma igualitaria entre zozoantelados y zozoantelados, lo que es súper interesante porque tenderíamos a pensar que los corales, o sea que las especies que nos faltan deberían ser de ambientes, por ejemplo, profundos, donde es más difícil colectar. Pero no, vemos que se divide en forma igualitaria. Y bueno, cuando vemos un análisis geográfico de la cantidad de especies, cada cuadradito representa el número de especies y cuando tiende hacia el color amarillo es donde hay más especies registradas en las colecciones. Podemos ver esta gran cantidad de registros para el Triángulo del Coral, que es una zona que se ha descrito con una gran cantidad de especies, pero también vemos vacíos, por ejemplo, en el Mar Rojo, en la zona de Madagascar o también en el sur de China, donde deberíamos tener más especies representadas de lo que está descrito por la literatura. Y bueno, finalmente hicimos un análisis que tiene que ver con la completitud de los datos, donde cada uno de estos puntitos es una institución diferente y registramos cuán completo estaban viendo el año, si es que el registro tenía el año, y las coordenadas. Entonces, podemos ver que instituciones como, por ejemplo, el Queensland Museum tienen una completitud de datos súper alta, a diferencia de instituciones como el Museo de Historia Natural de Londres, que tiene un poco peor la completitud de los datos. Pero acá me gustaría destacar algunas instituciones como Conavio, que es de México, que también está en esta zona que es como de mejor calidad de datos, o como también el INPEMAR de acá de Colombia, que está en el punto con 100% de registros con años y con coordenadas. ¿Y por qué esto es tan importante? Porque estos son los datos, estos son los registros que nos van a permitir hacer, por ejemplo, análisis de modelamiento y otro tipo de estudios. Y bueno, finalmente quisiera comentar algunos problemas que detectamos en todo este proceso. Por una parte, que hubo muchos registros en los cuales no pudimos hacer el match con WORMS, que es la base de datos mundial de registros marinos, como también algunos registros que estaban clasificados como ejemplares preservados, siendo que deberían estar en la sección de fósiles. Y bueno, finalmente utilizamos la base de datos del Museo de Historia Natural de Londres para analizar cómo podemos mejorar sus datos, y nos dimos cuenta que haciendo, por ejemplo, georreferenciación retrospectiva, podríamos mejorar y subir este 23% de datos con coordenadas, porque tenemos las descripciones de las localidades. Podemos hacer lo mismo también con el año y poder mejorar este dataset desde este punto hasta un punto mucho mejor, con una completitud mejor de los datos. Y bueno, para concluir, es importante, me gustaría destacar que las colecciones de historia natural son irreemplazables, y que esto es particularmente interesante en la situación actual en la que estamos viendo un aumento exponencial del registro de observaciones, pero en la colecta de los ejemplares estamos viendo una baja. Entonces, bueno, me gustaría destacar eso, y también que es necesario realizar mantenimiento de las colecciones de historia natural y definir, o sea, este tipo de análisis nos permite definir estrategias para la futura digitalización que hay que realizar con las colecciones. Y eso, muchas gracias.
Okay. Hello, good afternoon. I want to articulate 4 ideas that you're probably very familiar with. So the first idea is that collections memory can actually fade, disappear. The ideal case in many collections is that you have everything completely located, geolocated preferably, and data are there, and so when you digitize this collection, you have all the data you need. But the harsh reality is something different. Data can be missing, can be difficult to read if you digitize things when some time has passed. Perhaps there is no memory in the department, or about who wrote that label, or who that person, which is given by a single last name, is actually. So before that happens, you need to recover that. And if a lot of time has happened, well, then you have to use techniques to try to recover that retrospectively in some way. So real life is not ideal, unfortunately. However, many collections are already digitized, and many mistakes have gone into those databases. I took 2 databases, not too large databases, one for a Tetris project on pollinators, another one from my own department, which was also part of the previous one. I'm talking about different data sets. One belongs to 5 different institutions, more than 30,000 specimens of insects, 700 taxa for a wide period of time. Another one with slightly less specimens from different groups, 900 taxa, and a narrower time frame. So I wanted to return the memory to these collections, and the data for those collections were already in the database. That was the problem. It was already database. Okay, what can we do? I was focusing on 2 things, locations and collectors. In the case of collectors, you have a number of collectors, a very large number of collectors, many of which you don't actually know who they are, because they only know by a single last name, for instance. So what we can do, we can take this list of collectors and try to normalize things, for instance, removing extra spaces, or recovering diacritical marks. In the case of Spanish, you do it in a sequence. You first apply one algorithm, then next algorithm, then next algorithm. You can try then to separate multiple ways to spell the same person, or try to normalize how to express 2 different collectors in the same way. You can renormalize the way to express the last name, comma, first name, etc. So everyone is done in the same way. And then you start sorting and sorting and sorting and finding duplicates, so potential duplicates, and remove them. You remove duplicates, or you merge actually duplicates, and try to get a single authoritative, say, list of actual people. But who are those people? You have to go to records that you might have in the case of the university. We could go to academic records, to field records, and try to match by date, by collection place, or by last name, who that person was. In the case of universities, the additional problem is that many persons would only contribute 1 or 2 specimens, so statistical techniques are out of the questions in many cases. Well, finally, you group things, and then you get a dictionary of names. Is this of names that actually correspond to actual people. Finally, we know who those people were. Okay? In many cases, but in many cases you don't. If you sort collections, you also find patterns, which in those patterns might be interesting. In this plot, each line is the specimens collected by one single collector over time. You might see that there are interesting patterns, because if you took distinct names, you have a high entropy, but if you actually identify those people, well, at least the entropy can be reduced, but not so much. Actually, we could reduce the entropy by only 20%. It doesn't seem much, but it might be enough if you take into account that some of those are responsible for a large number of specimens. And then, if you separate by institution, you also find patterns. In this case, the colors, they represent different collections from different institutions in Spain. And, well, it might seem a mess, but you can order them by institution, and you find something different. For instance, you can find this, that some institutions have collections extended over time. Some institutions might have much narrower collections. You can guess which collections, which institutions are those that have a very narrow sector. University collections, in which collection was included into some study program. Okay? So the program starts, you collect, the program ends, you don't collect. Even in the case of large collections, that also happens, that new legislation forbids you to collect from collecting, which is why the universities could not continue collection, because the new laws made it very difficult to do any collection at all. Okay? So let's order them by starting date of collections. Again, each line is one person. And you see that there is a clear pattern that simply tapers off very quickly at that time when the new legislation made collection in the field quite difficult to come by, because you needed a lot of permissions, and you couldn't go to the next province with your provincial permission and so forth. So everything. But then academic collections, again, were so different. Those are academic collections, very normal, were quite different from reference collections located in large museums, which are more constant, very much like Catalina said before in the case of corals. Okay? This is the largest museum in Spain, the Natural History Museum. So, can we recover things? Yes, we can do that, and by using all those techniques, sorting, recovering, deduplicating, and so forth, in this case, those are locations. One of the main ways to actually identify a person is to see who was collecting, which names were collected in this place, and doing that by collector's times, you find patterns that enable you to actually reduce the entropy. So finally, from 20 collectors who were fully identified, we came up to 21 to 22% of collectors whom we know by picture, last name, and passport number, whatever. But in the case of the actual material, it goes up to 80%, which is much better. Okay, so by working on those few collectors that provided a lot of data, we can actually move the knowledge or recover the memory from 50% to 80%. And some things, this is basically the statistics of this recovery exercise. We could get up to 10 different names for one single collector, and the entropy reduction, as I say, was not too much, 22%, but in terms of the specimens, it was large enough. Okay, there is a second way, a secondary aspect, which deals with locality. I want to go quite quickly over this. But in the case of locators, you have the same problem. This is one single location in Spain, that's the Escorial, which can be known under so many names in the collections that is almost staggering. Those are different names for the same, exactly the same place, okay? And they were already in the database. So you can analyze this again and find the sources of entropy, like variations of the same name, or misreading, typos, or misinterpretation. Reading handwritten labels is quite difficult, as we know. Language, they might change language according to different dialects or languages in the same country. Historical use changes, administrative combinations, legal changes of names. So along the history, you have so many sources of entropy that is almost defies comprehension. But if you can somehow handle all of that, you can also reduce entropy, which enables you to reduce the load for georeferencing. You don't have to georeferencing 10,000 localities, you can georeference 5,000. What we did was to follow basically the same thing and reduced the entropy by 43% and got 400 georeferencing hours saved. And that's it. Thank you.
So we have a question online. I don't know if we have time to answer.
30 seconds, really quick.
All right, all right. When doing this work, when doing this work, do you add an identifier to the collector to the collection management system? Have you used binomial frictionless data package for collector attribution?
I didn't use binomial, which would have been very useful for the large collections, but we could not use binomial because the most cases there were students who were only known by one single record, so they are not in binomial. It was absolutely it was impossible to use binomial in this case, unfortunately.
Okay, thank you very much.
Welcome.
Mashallah.
Oops, excuse me. I'm stealing your microphone. Sorry.
Yes, because the next speaker is you actually, so yeah, you need to be mindful.
Hello, I'm Ashley. Hang in there, we're nearly done. I'm a research associate at the Royal Botanic Gardens Kew, and my focus is on the digital curation and enhancement of already digitized specimen records. So georeferencing at Kew is a huge part of what we do, and it feeds into many of our outputs, from taxonomic research to species distribution modelling, through to IUCN Red List assessments, and they then feed into tropical important plant areas, which is the designation of a tropically important plant area which has had policy impacts and on-the-ground conservation action. So what is georeferencing? We're all aware of the three big components of a specimen record, so what it is, the taxonomy, when it was collected, and where it was collected. So georeferencing focuses mainly on the where and occasionally the when. And in doing this work, I've realised that all things point to the sacred text, which is the best practices guide. And that defines georeferencing as the process or product of interpreting locality description into a spatially mappable representation using a georeferencing method. Initially, I found that quite vague, but that's because there are many ways you can georeference a specimen. But at Kew, we use the point radius method. So you assign a coordinate to the record and a radius. So georeferencing at Kew has been happening since about 2006 in terms of a concentrated effort. And it's been happening on an ad hoc basis as well as that before 2006 and after. However, out of all of the specimen records on GBIF that have come out of the digitization project that Sidra has spoke about already, only 800,000 actually have coordinates. And on the board there is Kew's top 10 list of countries with coordinates. And that's not representative either of georeferencing efforts, because as you know, when you collect a specimen, you should be collecting the coordinates now, but that's not represented in those numbers. So as a broad overview, this is kind of what I've been working towards in terms of collating all of this legacy georeferencing work, and that started with manually locating and standardizing georeferencing files. So for an IUCN Red List assessment, a researcher or assessor will collate all of the known information on a species, and so we have folders upon folders of this data. And so the Digi team kindly lent me 4 QA officers, and they went through all of Q's folders and identified the best georeferencing file and standardized the name for me. And I'm now currently in the consolidating and cleaning phase, so pulling all of this data together into one space and cleaning it ready for publication. But the bit that gets me excited is, as this work's predated digitization, these records don't necessarily have barcodes, so linking what is considered an enhancement to those records and making them above that MIDS2 and enhancing those records is really exciting. And then also being able to link those with the duplicate specimens from other institutions. So for a herbarium specimen, you get between 3 and 6 duplicate specimens for your one specimen. And I'll go on to what I'm doing later on in that. So this data set I'm really hoping to utilize to identify recurrent localities, tropical localities that may not be already available in online gazetteers, and to also compare and assess georeferencing tools. So currently at Q, we manually georeference our records. And that's not just necessarily Q records either. So I'd like to look at what tools are already out there that we can implement into Q's workflows to speed up that process. But also something that I find quite interesting is Q's records don't do too well on GBIS related records function. And so these records, which I would consider quite full, I'd like to use to improve Q's herbarium specimens to make those links between those duplicates so that we don't have duplicated effort in georeferencing later on. So as a bit of a side hustle to this legacy project, I've co-coordinated a georeferencing working group at Q, of which we'd like to establish an internal standard. So as I mentioned, georeferencing is an ad hoc thing at Q as well as a concentrated thing. And so because of that, the standards that we georeference our records to are different, and we record very different things. So that's one thing I'd like to establish. And then also, we have a new collections management system, and although a lot of our workflows have adapted to this new digital collection, georeferencing hasn't got that yet, doesn't have that yet. So I'd like to work on the workflows so that when someone is georeferencing a specimen, a Q specimen, they are able to get that into our management system, so that can then make it into GBIF and be available for other people. And also a big part of this this project is engaging with other institutions who have already done georeferencing projects. They will know better than what we do on how to do it at a larger scale. But also, I'm hoping by looking back at legacy georeferencing records, we can use that to look forward and georeference a significant part of, if not all, of Q's herbarium specimens. So I'm relatively new in this role, but I'd like to thank all of the people on the screen because they've put up with a lot of my questions and emails, and no doubt I will continue to come your way. So thank you.